Hi everyone, this is your Chapter 3 recording on cells. So as always, there'll be some learning objectives in the beginning of the presentation that you'll want to go back and make sure that you get. These are the same learning objectives that you can find in your textbook. So we'll start with talking a little bit about cell theory. Cell theory states that cells represent the basic units of life and that these tiny fluid-filled sacs house components that are responsible for all of the biochemical reactions that are necessary to keep an organism alive and allow them to survive. The primary function of each cell is to contribute to homeostasis, and the way that they do that is to make proteins. So when we look at a cell, we look at the basic parts to begin with. So we have three different areas that we're going to be looking in. We're going to look at this plasma membrane, which is the structure that keeps the outside out and the inside in. Then we'll look at the cytoplasm and its multitude of organelles, and then we will talk about the nucleus. So the cell membrane is the first thing, and I'm going to come back to the cell membrane later in this lecture, but we want to look at the structure of the cell membrane right now. So it consists of two layers of phospholipids, what we call a bilayer. And so looking at that molecule over on your right, you have the phosphate molecule, which is the round circular thing that you see. And then you see the two tails, and the tails are the fatty acids. So that's where we get the name phospholipid, because it's a phosphorus and a lipid put together. The head is hydrophilic, meaning that it likes water, and the tail is hydrophobic, meaning that it doesn't like water. So this cell membrane is what separates the inside world, the intracellular fluid, or the ICF, from the outside world, which is called the extracellular fluid, or the ECF. And so we have two layers that are with the heads that are hydrophobic, facing the watery world outside and the watery world inside, and then the tails in the middle. So if you look at that, it kind of looks like a balloon with two streamers. And so you got two layers of that. So phospholipids make up approximately 70 to 75% of the cell mass. The rest of the cell membrane is made of glycolipids and cholesterol. So what are some of the functions of the plasma or cell membrane? It serves as a mechanical barrier. It is selectively permeable. It is an it contains or possesses an electrochemical gradient. It's responsible for communication and cell signaling. So there are two types of proteins that are found embedded in that membrane. The first one that we talk about is called an integral protein. This is a protein that spans the entire width of the membrane, and it's referred to as being transmembrane. So channel proteins are an example of an integral protein. These selectively allow certain materials like ions to pass in and out of the cell. And then we have peripheral proteins. These are going to be found either on the outer surface or the inner surface of the lipid bilayer, but they don't span the entire width of the membrane. So taking a look at this, so we have our phospholipid bilayer. You can see that. You see the little phospholipids, the two layers of those, and then all of those blue structures that are either in the membrane all the way through or on one side or the other. So the one closest to the left is an example of an integral protein, the one that goes all the way through from one side to the other. And then you've got some next to it, a peripheral protein that's just on one side. So let's look a little bit more at some of these functions. Transportation, cell recognition, identification, receptors, ligands act as anchors, serve as enzymes, they form joining areas. So let's look at how they transport things. So I mentioned that the cell membrane or the plasma membrane is selectively permeable, meaning that it's going to allow some substances in and some substances it can't go through. So the membrane itself, only small molecules can move across the lipid bilayer. So things like oxygen and carbon dioxide gases and alcohol. There's two general methods that substances are going to use to pass through the actual membrane. One is a passive process. This means that no energy is required, and the other are active processes that mean that energy is required. So we're going to be looking at passive processes, including simple diffusion, facilitated diffusion, and osmosis, and then active transport, including endocytosis and exocytosis. So the way that these two sets of information are categorized is based on energy use. Either you use energy or you don't use energy for this process. So let's start with talking about passive membrane transport. The molecules or the ions are going to spread from an area of high concentration to an area of low concentration. And this just happens naturally. If you have a cluster of ions or molecules in a particular area, they are just going to naturally move themselves from an area of high concentration to an area of low concentration. And that process is the difference between the area of the high and the area of the low is called the concentration gradient, and this happens naturally. So there's no energy required for this to happen. Specifically, we can talk about diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration, no energy input. So if we took a tumbler of and we put a drop of food coloring in it and we just let it sit there, we didn't stir it, we didn't do anything, and we came back 5 or 10 minutes later, we'd notice that the entire container was all the color of the food coloring. And that's because the molecules that were in the food coloring are always in motion, even though we can't see that motion, and they're bumping into each other and they're bumping into the sides of the container, and they continue to do that till they space themselves evenly out. So diffusion can happen through a plasma membrane. So we move from an area of high concentration to an area of low concentration. And this is how those gases I mentioned earlier, like oxygen and carbon dioxide, can move across the plasma membrane. And so we have small nonpolar molecules in our picture, the little blue little stop signy looking things. Have more of those on the outside area where we have the extracellular fluid than we do on the inside. And then over time, which is where our arrow is pointing, over time, we notice that those are going to pass through that membrane from an area of high concentration to an area of low concentration. The ability for this to do this does not require energy, as I've said before, so we refer to it as being passive. Any damage to that plasma membrane leads to disruption in this ability of the cell to be able to do this. So that's molecules that can move from one side to the other without any help. But what if you are a molecule that can't get from one side to the other on your own? In that case, they can utilize what's called facilitated diffusion. So some substances can't get through that lipid bilayer because of their size or their charge or their polarity. So they're going to get a helper molecule. And that's what a facilitator does. A facilitator is a helper. And so facilitated diffusion means that we're going to have a molecule that's going to receive some help to get from one side to the other. And the example of this is going to be embedded protein channels. These are found in the membrane itself and carrier proteins as well, and they are going to allow larger molecules, usually polar molecules, to pass through those passageways. So they help or facilitate. I always think about it like a kiddo trying to get from one side of the street to the other near the school, and they have a crossing guard. We wouldn't want that individual to do that by themselves, that child to do that by themselves, or they can't do that by themselves, but they certainly can with the help of a crossing guard. All right, so let's look at a channel protein first. So a channel protein, you can see your phospholipid bilayer. You can see the outside world of the cell at the top and the inside world of the cell at the bottom. And you can see that you have this channel protein, the blue thing. And what it does is it allows molecules to pass through. They can't go through the membrane for whatever reason. They can't just pass through it. They're going to go through that channel. Some of those channels stay open all the time. Others are open because of chemical or electrical signals. Then we have carrier proteins. Carrier proteins are a little bit more selective. They usually are only allowing one type of molecule to pass over. The channel proteins, lots of different things can go through that channel. Carriers are a more specific type of facilitator. The diffusion rate in this case is going to be affected by the number of available carriers because they can't just simply float through like we saw with the channel protein. So sugars and amino acids, sodium ion, potassium ion, calcium ion, all those kinds of ions like that are transported through this mechanism. So let's take a look at that. You see that you have your outside world of the cell, and you have your plasma membrane, and then you have the inside world of the cell, and you have these carrier proteins. And notice that they're only able to fit the molecules that have a certain shape. And so in this particular case, they're going to pull it in from the inside, it's going to pass through, and then it's going to be dumped off into the, pull it in from the outside, and it's going to be dumped off on the inside. If we're talking about a very specific type of diffusion that involves water, then we talk about osmosis. So osmosis is specifically the diffusion of water through a semi-permeable membrane down its concentration gradient. So they're showing you that in the picture. You have your semi-permeable membrane, and you have water on one side, and as that water moves over to the other side, notice that the volume increases. All right, the osmolarity of a solution is the concentration of solutes that are found in the solvent, and I'll be talking about that in just a few minutes. When the solute concentration goes up, we say it has high osmolarity, and the water concentration goes down, and vice versa. And water is going to move by osmosis from an area of low solute or high water concentration to an area of low water. So water is going to move from an area of high to an area of low. So solutions, I've used these two words already, so let's kind of clarify that. Solutions are a homogeneous mixture when one material is dissolving another. And so the solute is what's being dissolved, and the solvent is what's doing the dissolving. And the classic example of this, of course, is Kool-Aid. So you have a Kool-Aid sugar mix that you get at the store, and that's your solute. And then you have the water, which is your solvent. And then when we mix those two things together, we dissolve the solute into the solvent, and we produce Kool-Aid. So let's talk a little bit about tonicity and solutions. So osmosis occurs when there is an imbalance of solutes outside of the cell versus inside of the cell. And so the thing that makes it a little challenging is that the solutes are different inside and outside, but it's the water that's moving. And so we name our solutions based on our solute concentration. So what we're dissolving, but it's the water that's moving. So that's always a little bit of a challenge to think about. So we have three different ways that we can identify our solutions. We can refer to them as isotonic, which means that the solute concentration on both sides of the membrane are the same. We have hypertonic solutions. Solutions are highly concentrated solutions, and we have hypotonic solutions, which are very dilute solutions. So let's talk about an isotonic solution for just a second. In isotonic, the osmolarity inside and outside of the cell is the same. So for every one water molecule, because remember, it's the water that's moving, the one water molecule that moves in, one water molecule is going to move out, and we call that net exchange. So remember that molecules are always in motion, and the term for that is Brownian movement. So they're not stationary, they're always moving, and that's what they're showing you in that bottom picture. Even though they are the same, the amount of water and the amount of solute is the same on both sides of the membrane, the water molecule is still moving in and out, but they're doing it equally. And so if we look at our cells underneath of a microscope, they look exactly the same. In a hypertonic situation, the cell outside of the cell has a higher osmolarity than the inside, so the water leaves the cell, resulting in the cell shrinking or crenulation. So I always say the cell gets skinny. So if you look at your picture of your blood cells over on the right-hand side in a hypertonic solution, that water is going to move out of the cell, and the cell is going to shrink down. If we put our cell in a hypotonic solution, the outside of the cell has lower osmolarity than inside the cell, and so water is going to move into the cell, resulting in cell swelling, which can lead, it doesn't necessarily mean it will, but it can lead to the cell rupturing or bursting, which is called lysis. And if you look at your picture, that's what you see. You have your red blood cell that's been put in a hypotonic solution, and when you put it into the hypotonic solution, water moves into the cell, and the cell bloats up. So why is this concept of osmosis so important in medicine? This is the basis for IVs. right? So how do I remember which way the water is moving in and out of my cells in these different solutions? It's going to sound silly, but this is how I remembered it. So in a hypertonic solution, my cell gets skinny, and I'm reminded that hyperactive people tend to be really thin because they're moving around all the time, right? And so I equate that to mean that in hypertonic solution, my cell gets skinny. If you don't like that example, I think about hypotonic solution. And in hypotonic solution, I think of the animal a hippo. Hippos are big and round and bloated looking, right? And so I remember that when I put my cell in hypotonic solution, it's going to get big and round. So if that helps, I hope it helps you. So these were all passive processes we're talking about. All of these things that we just talked about are moving molecules um, over a membrane without any energy required. It's just a natural process that happens. If we want to move things against their concentration gradient, then we have to put energy in, and that's referred to as being active transport. So we're going to talk about two different ways this happens: active transport through a pump and then vesicular transport. So membranes have lots of things in them, but they can also have something called a pump, and that pump is responsible for moving things against the concentration gradient, either from the outside to the inside or the inside to the outside. And the classic example of this, the pump that we know the most about in anatomy and physiology, is the sodium potassium pump. And so the sodium potassium pump is illustrated on this particular picture. You've got more sodium outside than you do inside. And so sodium wants to move into the cell, and you have more potassium inside than you have outside. So potassium wants to move out. But unfortunately, we need it the other way out. We need the sodium to stay outside, and we need the potassium to stay inside. And so these sodium potassium pumps are working against the concentration gradient and moving 3 sodiums out for every 2 potassiums that are moved in. And that's what they're showing you. If you look at the first pump on the left-hand side, it's got 3 sodiums locked into it. Then it opens to the outside world and it kicks those 3 sodiums out. Then it picks up 2 potassiums and brings them in and dumps them back in. All right, so it's maintaining those gradients that it needs by actively uh, uh, working against the concentration gradient. So now let's talk about vesicular transport. So if you remember from our discussions before, a vesicle is a package full of stuff, right? Whatever it is, it's something that we need to move. When we talk about vesicular transport, we're not involving any type of membrane carriers or channels or anything like this. This is how things that are brought into and out of the cell without using those types of structures. Large particles are usually what we're talking about here, or liquids. This does require ATP, and the two examples we'll be talking about here are endocytosis and exocytosis. Endocytosis means to bring into the cell, so endo means into, cy means the cell, osis means the process of. So the process of bringing something into the cell, and then exocytosis, as you can imagine, is the process of transporting something out of the cell. So we'll start with endocytosis. So this is how we ingest material by bringing it into the cell. There's 3 different types of endocytosis that we're going to talk about. We're going to talk about phagocytosis, pinocytosis, and receptor-mediated endocytosis. And again, a vesicle is a membranous sac. It is spherical and hollow. It's a spherical hollow organelle that's bound by or covered in lipid bilayer membrane. So in phagocytosis, this is how it works. Basically, you see your cell membrane. All right, you see the outside world that's blue. There's the inside of the cell, and there's the hot tamale that the cell decides that it needs. All right, and so what it's going to do is it's going to push its cytoplasm from the inside of the cell up against the inside of the membrane, and it's going to force these like arm-like structures to come out and surround that hot tamale. And then it's going to enclose it and bring it in. And when it does that, notice that the vesicle is now made of the cell membrane, the little chunk of cell membrane that was involved in making those arms and pulling it in. And so that is what phagocytosis is all about. right? So they refer to those as false feet, the pseudopodium that you make. To me, they look like arms that just reach out and grab it. Pinocytosis is how do we bring in a liquid? We can't reach out and grab liquid. It doesn't work the same way. So instead, what we do is in the cell membrane, if you look at your little picture there, the gold line is the cell membrane. We make a little divot or a little dip in that, and the whatever we're looking for, the liquid stuff, it just falls in, right? And then it pinches off and brings it in. Again, it's surrounded in cell membrane. But that process of bringing things in through pinocytosis or phagocytosis, they're not especially specific. So if we want one particular molecule, we're going to do something called receptor-mediated endocytosis. And in this process, the cell's going to create that dip again in the outer membrane, and that area is going to fill. But what's going to be filled in that area, there's going to be receptors. So receptors are like parking spots for a particular molecule, and it's going to bind to that receptor, and then once all of those parking spots are full, it's going to close off and bring it in. And so receptor-mediated endocytosis allows for specificity of what we're bringing into the cell. So let's talk about how do we get stuff out. So the cell makes things, hormones, it makes neurotransmitter, it makes all kinds of different things depending on the cell. And those things might need to be exported out of the cell to another cell, or it may have waste products. Cells are like factories. They make waste. That waste might need to be removed. Whatever it is that we want to get rid of, we're going to engage in a process called exocytosis. So we're going to have our vesicle that's wrapped in a little piece of cell membrane. We're going to merge that vesicle with the actual cell membrane, which is orange on our picture there, and then we're just going to dump that content out into the extracellular space. Okay, so endocytosis is bringing things in, and we pinch off a piece of the cell membrane. Exocytosis is kicking things out, and we wrap a piece of cell membrane and add it to the membrane. So these two processes, endocytosis and exocytosis, they're what keep the cell the same size. Because every time we pinch off, we're taking some away, but every time we put things out, we are adding to it. So those two processes are maintaining the size of our cells. A little bit about resting membrane potential, RMP. We're going to come back to this idea in muscle. But I just want to get it out there because it's important for some cells. So the resting membrane potential is the electrical potential difference across the cell membrane when the cell is just sitting there minding its own business, not actively transmitting signals. So cells transmit electrical signals, and when they do that, they're using this idea of the resting membrane potential. If we were to measure that, we have our little probe there. If we were to measure our resting membrane potential of our neuron, in this case, it would be a roughly negative 70 millivolts. So make sure you've got that negative in front of there. At rest, when we're doing the resting membrane potential here, at rest, the inside is more negative than the outside. So take a good look at that picture, and you can see on the cytosol side, the bottom side, there's little negative signs, and on the extracellular side, there's little positive signs. So when the membrane is at rest, it's not at 0. It's at negative 70, and that'll become important when we talk about how signals are transmitted in later chapters. So it's time to go on a tour of a typical eukaryotic cell. That's what we're studying this semester. So we're going to take a tour of that and learn all of the different parts. So I've used the word eukaryote. Let's talk about what a eukaryote cell is first. There's two ways to be a cell. You can be a prokaryotic cell. This is a simple basic cell that lacks any membrane-bound nuclei. In other words, the nucleus is not compartmentalized by a special structure, and it doesn't really have a lot of membrane-bound organelles like the ones we'll be talking about in a few minutes. Prokaryotes are the topic of microbiology, and that's not really the topic of our focus. That's not our focus in this particular course. So if you have to take microbiology after you finish 2 semesters of anatomy, we'll be talking about prokaryotic cells in there. In this course, we're focusing on eukaryotic cells or eukaryotes. These cells are more complex. Their nucleus is bound by a membrane, so it's compartmentalized or kept away from the rest of the organelles, and some of the organelles have their own membrane as well. Regardless of the type of cell, the primary function of all cells is to make protein. If someone had told me that in the very beginning of my study of biology 40 years ago, I would have been so much better off because I didn't understand that. Now that I understand that completely, that cells make protein, and this whole thing that we're going to be talking about ultimately is about how the cell makes protein, that would have been something that was a lot clearer to me. So prokaryote versus eukaryotic cells. Prokaryotes, what we do in microbiology. Eukaryotes, what we're focusing on in anatomy and physiology. Just because it's a simpler cell, prokaryote cell doesn't mean it's not successful. Prokaryote cells have been around much, much longer than eukaryotic cells. But nonetheless, they are a easier cell to study because they don't have quite as many fancy organelles that we're going to talk about. So I mentioned I was going to come back to the cell membrane, and here I am back at the cell membrane. So we were talking a little bit about what the cell membrane looks like and mostly what it does in the first half of this lecture. Now we're really going to focus on what it looks like and get a little bit more detail of that. So the cell membrane and the plasma membrane are the same thing, and I've been using those terms interchangeably. There's another term that you could actually use. It's called the plasma lemma. That's an older term, and you won't see it in textbooks very often anymore. However, some um, structures for specific cells use part of that name. And so let me give you an example. When we study nervous cells, neurons, part of the cell membrane of a neuron is called the neural lemma. So they use that plasma lemma idea. So that's why I like to throw it out there. You'll refer to it as a cell membrane or a plasma membrane. What you can't refer to it is a cell wall. You do not have any cell walls, right? Cell walls are bacteria and plants and fungi, and you are none of those things. You are a human being, and you don't have any cell walls within your body. So the cell membrane, as I've mentioned before, its job is to compartmentalize the cell or separate it from the outside world. So we use these six words. Is it five or six? 6 words to describe a cell membrane. And it sounds really complex, but we're going to break it down. So if I were asked you to characterize the cell membrane, what you would tell me is that it's selectively permeable, fluid mosaic phospholipid bilayer. That sounds really complicated, but let's just talk about it for a second. Selectively permeable or semi-permeable, so either 5 or 6 words, means that it's picky. It's not going to allow just everything to come into the cell. And we just saw that with those channel proteins and those different types of membrane transport we were talking about. So it's selectively permeable. Fluid. The cell membrane is not a solid structure. We look at it on a picture and it looks like it's an encased solid structure like a shell, and it's not. It is fluid. It's really nothing more than phospholipids, and phospholipids are oil. So it's basically an oil ring around the cell. But the fact that it's fluid means that it's wiggly. All right. That's a big, fancy, important biology term there, wiggly. Mosaic. Mosaic means that it has other things embedded in it, like proteins. And you just saw that with all the integral proteins that were embedded in the cell membrane itself. Phospholipid is describing the type of molecule that makes the membrane. And then bilayer, you've seen multiple times now that there's 2 layers, and that those 2 layers are arranged that the phosphate head, the hydrophobic portion, is arranged towards the outside and the inside of the cell, and the tails are in the middle. So it's kind of like a cell membrane sandwich. Your 2 pieces of bread are your phospholipid heads, and then the tails are your peanut butter and jelly. So there's a picture of it again, the selectively permeable fluid mosaic phospholipid bilayer. Notice how it looks kind of wavy. They're trying to represent in a static drawing that it's wiggly, right? Again, a picture of that phospholipid molecule. So let's talk about our cytoplasm. So everything inside of the cell membrane, but outside of the nucleus. So that space in between, right? That's what we're looking at here. It is filled with a jelly-like substance that's called the cytosol. And it's the cytosol that is the medium where all of our biochemical reactions are going to happen. It's filled with something called organelles, which in Latin means little organs. These are structures that perform different functions within the cell. So when we take the cytosol and the organelles, we put them together, we create cytoplasm. So cytoplasm reminds me of Jello with fruit in it. The Jello would be your cytosol, and the organelles floating around would be your fruit. So let's talk about organelles. Some of them have a membrane that separates them from the rest of the inside of the cell, and so we refer to those as membranous organelles. So things like our mitochondria and our Golgi apparatus are examples of that. And then others are not compartmentalized. They don't have a specialized membrane that's encasing them. Some of the filaments and the centrosomes we'll be talking about will be examples of this. So a cell is a busy place. And as I said, what does a cell do for a living? It makes protein. It makes protein. Make sure you understand that. It makes protein. It's just like a factory, right? So just like a factory, you have different individuals that are doing different parts of the assembly process to make something. And that's exactly what's happening with our cell, is it's making proteins and other substances, but specifically, mostly proteins. And there are organelles, each of them has a specific job related to that function. Now let's talk about the nucleus for just a second. So this is considered the control center of the cell. It stores all of the genetic instructions in the form of DNA for making protein. So it is the instruction manual for making all of the proteins that the organelles are going to make out in the cytoplasm. Most cells only have a single nucleus, but there's some notable exceptions. Muscle cells, as we'll see in a few chapters, have more than one nucleus, so they're referred to as being multinucleated. Red blood cells lose their nucleus during the maturation process and are referred to as anucleated or enucleated. So when we look at the nucleus, it's the whole structure is called the nucleus, and it's surrounded by its own membrane called the nuclear envelope or the nuclear membrane. And it has little openings in it that are called the nuclear pores. And so they're showing you that. They're kind of a gold color on that picture. Those are the passageways for things to move in and out of the nucleus. So the DNA can never leave the nucleus, but it has the instructions. So how in the heck are we going to get that information out to the organelles that are going to make that need those instructions to make the protein? Those instructions are going to move out those nuclear pores. The nucleolus is a region in the nucleus that's responsible for making the ribonucleic acid or RNA that's necessary to construct the ribosomes. So that dark purple area you see, that's the nucleolus. There's usually one, there's sometimes two, so it's nucleoli in Latin, and I at the end of the word means it's plural. Chromatin. So inside of the nucleus are threads of chromatin. Chromatin is made of DNA and associated proteins. When we condense all of that chromatin down, we form chromosomes. So that's the difference between chromatin and chromosomes. So chromatin is the uncondensed form of a chromosome. So a chromosome, as we've already talked about a little bit, is all of the DNA, and it's got proteins in it called histones. Humans have approximately 22,000 genes. A gene is a individual instruction set for building a specific protein. So your body knows how to make 22,000 different genes or proteins. Those genes are found distributed on chromosomes. So they're clustered together on 23 pairs of chromosomes or 46 total. So you have one chromosome you got from mom and one chromosome you got from dad. They have the same information. There may be variations in that. So, like for instance, let's just say hypothetically it's for eye color, which is not really how eye color works, but we're going to pretend it does. And so you get one chromosome from dad for blue eyes and one gene from dad for blue eyes and one gene from mom for brown eyes. And that information is on the same chromosome. So we say 23 pairs because they have the same information on them. Or you could say 46. but you don't have 46 pairs. So either 23 pairs or 46. The number of chromosomes you have is unique to your species. So I can't remember the exact number, but it's kind of a crazy number. Like shrimp have like 119 chromosomes or something like that. So it doesn't have to do with the complexity of the organism. It has nothing to do with that. It just is. So you and I have 23 pairs of chromosomes. All right, the endoplasmic reticulum, the ER. This is a system of channelways that is continuous with the nuclear membrane or envelope, and it's composed of the same material as the lipid bilayer. So I want you to think about it like this. Let's pretend for a second that you have a plant that makes widgets up in Columbia, South Carolina, and you want to get that widget after it's manufactured down to Charleston to put it on a ship. But there's not a highway. So the only way for you to get things from Columbia down to Charleston is to put it on the river and float it on down. Well, that's what the endoplasmic reticulum is like. The endoplasmic reticulum is like a waterway. And so as proteins are made, they're put into that waterway and they are channeled down to the next organelle. So what the ER does is it serves as a passageway throughout much of the cell, and it functions in transporting, synthesizing, and storing materials. There's two ways to be an endoplasmic reticulum. You could be the rough endoplasmic reticulum, which means that you have ribosomes attached to you, and as those ribosomes make proteins, because that's what ribosomes do, they dump them into that waterway and they're taken down to the next organelle. It's called rough because the ribosomes attached to it give it kind of a bumpy appearance. So as I said, ribosomes are going to make protein, then they export that protein into the ER, and that's transported down to the next destination. Or you can be smooth ER. So if rough ER means you have ribosomes attached, then smooth ER means you don't. So smooth endoplasmic reticulum is the one organelle that has a variety of different functions depending upon the cell that it's located in. Typically, different organelles, they do the same thing regardless of the type of cell that they're in. So if they're in a muscle cell, they do the same thing as if they're in a liver cell, and the same thing if they're in a stomach cell. It doesn't matter. But the smooth ER kind of is cell dependent. So it has some overall functions that it always does, but it also does some extra things in certain cells. So it's a little bit different than the rest of them. All right, so it has a variety of tasks that it forms. It performs. It is responsible for the synthesis of lipids, phospholipids, and steroid hormones, and it does that pretty much wherever it is. But it also, in certain places, it can store and regulate the concentration of cellular calcium ion. That's in nervous cells and muscle cells. And it also can act as a detoxifier, like in liver cells. It can metabolize some carbohydrates. So depending on the cell it's in, it can actually do a few things different than smooth endoplasmic reticulum in another cell. The Golgi apparatus, also known as the Golgi body. So Golgi is somebody's name, Dr. Golgi. And so when you write out Golgi apparatus, you would want to capitalize Golgi because just the G in Golgi, since we do that with somebody's proper name. This is the packaging and shipping center or the UPS store of your cell. So it's going to be receiving all the products that are being made, mostly proteins, but there's a few other things. It's going to receive those, it's going to sort them, modify them, and ship them off to wherever they need to go. Maybe they need to go to a different part of the cell. Maybe they need to be exported from the cell. It's just the shipping center. It also produces new lysosomes. Lysosomes and peroxisomes. So our cell as a factory is kind of a dirty place. It makes a lot of waste product. And so that is going to be the function of the lysosome and peroxisomes. The lysosomes contain digestive enzymes and they break down organic compounds. These organic compounds sometimes can be nutrients that have been brought in through phagocytosis. It also can be defective proteins. So if you remember back to chapter 2 when we were talking about proteins and the 4 different levels of protein structure, sometimes they don't get folded correctly, and if they don't get folded correctly, a lysosome will take them apart and recycle the amino acids from that. A bacteria, we saw that with phagocytosis when we were looking at the red tamale. Let's just say that red tamale was an E. coli bacteria, right? And so the lysosomes can destroy that. Anything that needs to be broken down or recycled is taken care of by the lysosomes. The peroxisomes are a little bit more specialized. They're a specialized form of a lysosome that deal mainly with toxins, and so their main function would be to serve as a detoxifying center. They primarily are located in the liver because the liver is what detoxifies most things in our body. And then ribosomes. I've talked about ribosomes already a couple of times in brief, but let's talk about it in a formal way. This is an organelle that is the site of protein synthesis. So this is the organelle that's probably the most important when we talk about all this, because what does a cell do for a living? It makes protein. And where does that protein get made? It gets made on a ribosome. So a ribosome is made of two subunits, a large and a small subunit, and each of them is responsible for the process of transcription and translation of the DNA to make the protein. So I'll be talking about that near the end of the lecture. Ribosomes can be free floating within the cell, or they can be bound to that endoplasmic reticulum that we looked at a few minutes ago, and they can go back and forth between those two states. The mitochondria, I think most people remember the mitochondria from their high school biology or middle school biology. The mighty mitochondria is an oval-shaped organelle. It is the energy transformer, sometimes referred to as the powerhouse of the cell. The process of cellular respiration that we learn about next semester is the conversion of nutrients into energy, and that energy that's converted from our glucose is stored in the form of ATP in the mitochondria. All right, so the muscle and neuron cells that are highly energetic, they're going to have loads and loads of mitochondria. Cells that aren't so energetic, like maybe a fat cell, an adipocyte, they might have fewer mitochondria. Centrioles are barrel-shaped microtubule structures typically found in pairs. They look like two pieces of rigatoni pasta to me. They organize the cytoskeleton that we'll be talking about in a few minutes. They are responsible for producing the spindle as well in cell division. And so they are also important for assembling the cilia, which are extracellular attachments to some cells. So there's what your centrioles look like. So speaking of the cytoskeleton, here it is. The cytoskeleton is a fibrous network inside of the cell. The cell does not have bones, but it has what to me are like tent poles. So if you've ever put up a tent before, you know that you put poles inside of it to keep it from collapsing. And that's how the cytoskeleton works. There are this crisscrossed fibrous proteins all throughout the cell that help to keep the cell keep its cell shape, but it's also responsible for the motility of the cell if the cell moves, responsible for cell reproduction when we talk about the spindle apparatus in a few minutes, and for transportation of substances within cells. The larger filaments actually serve kind of like a monorail, like you have at Disney World, and can move things from one side of the cell to the other. There's three different types of cytoskeletal elements, and they are based on the diameter of the fiber. So the skinniest ones are called the microfilaments. The fattest ones are called the microtubules, and then right in between are the intermediate filaments. Cellular extensions, these are specialized structures that extend from the outside of the cell membrane of some cells. So we'll be talking about microvilli, cilia, and flagella. The microvilli are projections. They are actually foldings of the cell membrane. So imagine the cell membrane like a piece of elastic, and then I want you to squunch that up, and how it would all fold up on itself. That folding up on itself, that would be your microvilli. Right, so these are creating more surface area in places like the small intestine. So the fuzzy pink stuff you see on the very outside there, that is a microvilli. Cilia are not part of the cell membrane. These are hair-like structures. They're not hair, but they're hair-like structures that are on a lot of epithelial cells that line places like airways and fallopian tubes, places like that. The cilia move rhythmically. In other words, they all move together and they beat constantly. And so they're able to move things by the cell. So for the most part, our cells can't move, but we can move things by them. So I always say the cilia are kind of like, if you've ever been in your bathtub and you had the bathtub on a certain temperature, and then after a little while, it starts to get kind of cool in the bathtub, the water does. So you turn the hot water on and then you use your arms to swish the water around the tub. Your arms would be like the cilia. Flagella, the plural of this, flagellum, the plural of this is flagella. This is a larger structure than a cilium. It's much longer and it's specialized in cell locomotion. So it works kind of like the rudder on a boat. It's the only flagellated cell in the human body that we have is called